Stages of intramembranous ossification. Mesenchymal cells differentiate into osteoblasts and a network of collagen fibers appear between these cells. This site where the osteoblasts first appear are called the centers of ossification. Osteoblasts secrete organic intercellular substance. Some cells are surrounded by the matrix, forming the lacunae, where they are converted into osteocytes. Osteoblasts proliferate and differentiate in a radiating manner from the center of ossification, forming the trabeculae. The trabeculae join to form the cancellous bone. The spaces between the trabeculae are occupied by blood capillaries. The osteoblasts secrete alkaline phosphatase in addition to the organic matrix, which calcifies the matrix at the site surface and ends. So, the new bone is deposited in the ends, which increase the length, and the site surface, which helps for the formation of lamellae, which in turn converts to compact bone. In fetal skull, there are frentinels between the sutures, which are closed within 2 to 18 months by the appositional method of growth. Most of the long bones in the body ossify in cartilages. Now let's see the stages of endochondral ossification. Cartilaginous model of the long bone consisting of hyaline cartilage is differentiated from the mesenchyme of the limb. This model is surrounded by a membrane known as perichondrium. Cartilage model grows in length by interstitial method and width by appositional method. Interstitial method takes place in the substance of the cartilage and the growth occurs near to the ends than in the midsection of the model. The cartilage cells are arranged in rows of longitudinal columns and are named from the end to the midsection as zone of proliferation by mitosis, zone of maturation of cartilaginous cells, zone of hypertrophy of the cells, and zone of calcification of the cartilage cells. The mature cells secrete the cartilaginous matrix around them. The hypertrophic cells secrete alkaline phosphatase, which precipitates the calcium salts in the matrix. The calcification of the matrix produces death of these cartilaginous cells due to lack of diffusion of nutritive material. The hypertrophic cells die and leave spaces around them known as the primary areolae in the midsection of the model. The perichondrium is vascularized and its inner layer is differentiated into the osteoblasts. The osteoblasts deposit new bone by appositional method around the center of the model as the periosteal collar. The perichondrium is now called the periosteum. The osteogenic layer of the periosteum forms the periosteal bud, which contains osteoclasts, osteoblasts, and blood vessels. The bud excavates passages through the newly formed bones and passes into the calcified matrix as the primary center of ossification. The perforation of the bud persists as the nutrient canal. The osteoclasts of the periosteal bud destroys the calcified walls of the primary areolae, cause fusion of the original cavities and form larger spaces known as the secondary areolae. This is filled with marrow and are lined by the osteoblast. The osteoblasts proliferate, deposit new bone on the walls of the secondary areolae and on the remains of the cartilage, forming cancellous type of bone. In long bones after birth, one or more secondary ossific centers appear at the ends of the cartilage model, forming the epiphysis. Ossification extends in all directions except at two areas. At the end of the model, which persists as the articular cartilage throughout life. Between epiphysis and diaphysis, the cartilaginous model persists as the epiphyseal cartilage, which persists as long as the bone grows in length. When growth is complete, the epiphyseal cartilage is replaced by bones. Once we know the stages of ossification, let's see the laws of ossification. Laws of ossification. The areas where the bone formation of ossification starts in the cartilaginous model are known as the centers of ossification. The primary center is that from which the main part of the bone is ossified. As a rule, the center appears before birth with some exceptions. The primary center of tarsal of carpal bones appear after birth except talus, cancellous and cuboid bones. The secondary center is that from which accessory part of bone is ossified. This center appears as a rule after birth except at the lower end of femur and sometimes upper end of tibia. Law of union of epiphysis. The law enunciates that the epiphyseal center, which appears first, unites last with diaphysis. Growing end of long bones. The growing end is situated against the direction of nutrient foramen of that bone. The dictum saying, to the elbow I go, from the knee I flee. Let's see the parts of young long bone. The following are the parts of the young long bone: diaphysis, epiphysis, epiphyseal cartilage and metaphysis. The diaphysis. It is developed from the primary center of ossification. The length of the diaphysis increases by interstitial growth of cells of the epiphyseal cartilage, followed by the deposition of bones at the metaphyseal ends of diaphysis. How does the cartilage grow? Epiphyseal cartilage and metaphysis constitute together the growing zone of a long bone. These are the following zones: zone of resting cartilage, zone of proliferating young cartilage cells arranged in rows of longitudinal columns, zone of mature cartilage secreting the alkaline phosphatase, and zone of calcified cartilage where the cells die in the calcified matrix. The metaphysis. The end of diaphysis facing towards the epiphyseal cartilage is known as the metaphysis. Do you know the importance of metaphysis? It is that it is the most actively growing area of a long bone. It has profuse blood supply. Muscles, ligaments and joint capsules are attached close to the metaphysis. A number of small parallel vessels enter the metaphysis and forms hairpin-like loops. Here, the blood gets stagnated. So, it is the most common site for osteomyelitis of bone in children. Next, we have the epiphysis and its types. Epiphysis. It develops from the secondary center of ossification. Pressure epiphysis. It transmits the body weight and protects the epiphyseal cartilage. Example, heads of humerus and femurs. Traction epiphysis. It is produced by the pull of some muscles. Example, trochanters of the femur and tubercles of the humerus. Next we have atavistic epiphysis. It is phylogenetically an independent bone and is attached secondarily on a host bone to receive nutrition from the host. Lastly, we have aberrant epiphysis. Example, base of the first metacarpal bone.